So we are live. I know we're at the top of the hour. People will begin to trickle on as always. So we'll just maybe give it 1 minute to make sure everything is set up correctly. Yeah. Shelley, are you going to be presenting slides today, or are we just going to go through the Q&A?
I have the same set of slides that you guys have from before. So if you have those, that I'll just walk through them. I think it's just 10 slides.
Yeah, hold on, let me grab those. Great, well, welcome everybody. While we wait to just pull up those slides, I'm Jessica Salzwedel. I'm a program manager here at AVAC, and we're joined by Dr. Shelley Karuna, who is the Director of Clinical Development at HVTN, the HIV Vaccine Trials Network. And you're joining us as part of a session for the Global Village, as part of the Research Literacy Networking Zone. For those joining on both YouTube and Zoom, this is part of a global session that is happening because we are unfortunately not able to be together for the AIDS 2020 conference. Obviously that conference was put on hold because of SARS-CoV-2, but perhaps the benefit is that we are able to get together in this space globally. Okay, here we go. How do I make these? So Shelley, I unfortunately don't have the slides as slides, but I only have them as a PDF. I can probably share my screen. Okay. This is why it's always good to do the HRM. Hopefully everybody can see the pieces on the screen. Great. Go ahead, Shelley.
Okay, super. Thank you, Jessica. It's really a pleasure to be here and talk about the AMP studies. I have just, I think it's 10 slides, so not too much, and there will be hopefully some time at the end for you all to ask any questions if you'd like. And my understanding is that this is really to give you all an overview of the studies as they wrap up, because the studies actually began in 2016. The work on them began in late 2014 and 2015. So we've been at the HIV Vaccine Trials Network and the HIV Prevention Trials Networks, the two networks that came together to design and implement these studies. We have been living in this for the last 5 years, but appreciate that many of you have not, and want to just set the stage really for the results of these studies as they come in just the next few months. If you want to move to the next slide or the next page, thank you. So the AMP studies, when they launched about 5 years ago, really were defining a new path forward in HIV prevention, evaluating a common idea, an idea that's been around for over a century, of assessing whether antibodies could be used to prevent HIV infection. They've been used for over a century in preventing other infectious diseases. You might have heard about them in the use against hepatitis, for example, but not in HIV until now, while we were testing that idea in these studies. AMP stands for antibody-mediated prevention. And if you go to the next slide, you'll see the exact antibody that we were evaluating in the AMP studies, an antibody with the oh so catchy name of BRCA1. It is a broadly neutralizing antibody, so it's a particularly special antibody that binds to a particular spot on the protein that is dotted around the HIV, the virion itself. It was actually, this antibody was discovered in an individual who had been living with HIV for over a decade without antiretroviral therapy and was controlling their HIV with no antiretroviral therapy. And the idea is that antibodies like BRCA1 may have contributed to that person's control of HIV without ART. This antibody was identified by and then brought to the lab and copied multiple times, made hundreds of antibodies like this by John Mascola and his colleagues at the Vaccine Research Center, a research center part of the National Institutes of Health. The study questions that this antibody is allowing us to ask and evaluate is first and foremost, as in all of our clinical trials, is this antibody safe? We monitor people really closely in this trial and to answer that question. And also, are people able to take the antibody without becoming too uncomfortable? Is it well tolerated? Those are two crucial questions to ask about any drug or medication or intervention that we want people to take. Next, back to the previous slide. The next question for the study is, does the antibody lower people's chances of acquiring HIV? And if it does, how much antibody is required to provide that protection from HIV? These are two crucial questions in any efficacy study. Does this intervention work? Does it accomplish what we hope to accomplish? And in this case, that's preventing HIV. All right, and then the next slide shows how the study was designed to be able to answer these questions. I said study singular, but actually there are two AMP studies. They are parallel studies. They're designed exactly the same way, but they're conducted in two unique populations. One in the green box represents the men who have sex with men and transgender individuals in North and South America and in Europe. So one of the studies was conducted among those individuals. And then in the orange box, the second study, the sister study, if you will, was conducted among women at risk of HIV acquisition throughout sub-Saharan Africa. Individuals in each of these studies were randomized to receive either the lower dose of the antibody, 10 milligrams per kilogram, the higher dose of the antibody, 30 milligrams per kilogram, or the control, which was just normal saline, like a placebo. And as you can see, among the, for example, 2,700 individuals enrolled in the Americas and Europe, 1/3 received the lower dose, another third received the higher dose, and another third received the control. And that same kind of division applied for the women in sub-Saharan Africa as well, for a total of about 4,600 individuals enrolled globally. Each of these participants, if you'll go to the next slide, you'll see each of these participants received 10 infusions. They received these infusions about every other month. And this shows the visits that these individuals came in to receive that infusion, that IV intravenous drip of the antibody or the placebo control. Now, between each of these visits, we saw individuals, participants, actually monthly. So while they would come in for an infusion every other month, they would actually come into the clinic every month. They would receive HIV testing, counseling, referrals for any other needs. They would answer questions, and we would check on their general health as well. The next slide actually shows where in the world these global studies were conducted. The green dots reflect sites where the study in MSM and transgender individuals was conducted, and the orange represent the sites where women contributed in sub-Saharan Africa to the AMP study there. The sites are actually listed on the next slide along with the enrollment numbers at each site. So you can see that enrollment was distributed quite globally There were certainly sites, for example, those in Lima that enrolled around 250 participants, other sites that enrolled fewer participants, as you see reflected here, truly a global trial. And the next slide reflects kind of across time how this trial unfolded. The first participants were enrolled in 2016 in each of these studies. The last participants were enrolled in 2018 in each of these studies. Really a remarkable accrual rate, remarkable enrollment pace in these studies, particularly appreciating that this really did ask a lot of these extraordinary volunteers to come into the clinic every month for a little over 2 years and to receive intravenous infusions or IV drips. And it was just remarkable, the enthusiasm that, that we saw and that our sites saw in the communities globally where these trials were conducted. I wanted to make a note here that, again, as we're in July 2020, we are looking to likely September 2020 when we would publicly be able to present the results. The analysis is going on right now. And these results will include the safety and tolerability data, and also, does this antibody prevent infection? And if so, how much antibody is required to prevent infection? We can actually provide those results in September because participants will have completed their infusions or have completed their infusions by now. We do, however, continue to follow participants for safety and tolerability even longer. So the final follow-up visits are actually going into early 2021. Even after we'll have the answer to those primary questions of, does this work? And if so, how much is required for HIV prevention? And last but not least, on the final slide, you'll see just what has been accomplished in the last several years. Over 40,000 infusions have been given in AMP, 4,625 individuals, remarkable individuals, enrolled in these trials and stayed enrolled in these trials. You'll see retention and adherence was really quite high in these studies, which I think really speaks volumes to the remarkable generosity of these clinical trials participants, their commitment and dedication to helping us learn if this novel tool can work to prevent HIV infection. Can we expand our toolbox for HIV prevention as we need to do, and offer people more choices for HIV prevention? So I am as eager as perhaps many of you are to learn what the results will tell us in September. I don't know what they will tell us, but I am waiting with bated breath. I'm happy to answer any questions that you may have about the studies. Jessica, I think you're on mute.
I was on mute.
Okay, great.
Yeah, so thank you so much for that really wonderful overview. And I do want to make sure everybody remembers this is part of the Research Literacy Networking Zone as part of the Global Village at AIDS 2020. So we do want to make this participatory, whether you're streaming here on Zoom or live on our YouTube channel. Please feel free to ask questions and we can ask them to Dr. Karuna. But I want to start off with perhaps maybe a simple question, but you mentioned the study design. It didn't have a lead-in, and we know that broadly neutralizing antibodies are something that stay in the body. And in the cabotegravir data that was just presented at AIDS 2020, we saw that there was a 1-month oral lead-in for the long-acting portion. Can you talk a little bit about why there isn't a lead-in period for the broadly neutralizing antibodies?
Right. So in the cabotegravir study, for example, they had an oral version of a very similar drug that they were able to provide before giving the injectable version of that drug, which is the injectable version is the version that was designed to last much longer in the body. There is no oral antibody for BRCA1, so that wasn't actually even an option. But there were hundreds of intravenous infusions of this same antibody including at the doses that are used in this study, that are used in this study, that were given to participants in earlier phase trials prior to the launch of the AMP studies. So we did have that baseline safety data in individuals in phase 1 trials that were conducted prior to the AMP studies. Also, I would say that these antibodies differ from other drugs in that they are fully human. So you can think of them as kind of natural products. We all have antibodies circulating throughout our body ordinarily, and this is one more antibody, very similar to those that are naturally made by our body and circulating throughout our bodies. So we were encouraged by the really outstanding safety profile that we saw in the earlier phase studies, and we watched participants very closely in this study, and the safety data will be part of what we're able to share in September.
Great, no, thank you. That's a really helpful clarifying piece. So one of the questions, obviously, as we prepare and we wait for these results, we're often thinking about the future, what comes next, especially because BRCA1 right now we know is not going to be moving into licensure. So can you talk a little bit about perhaps the future of broadly neutralizing antibodies, particularly the cocktail or the multiple broadly neutralizing antibodies that we've heard throughout the week?
Yes, I'm happy to. So I guess I think of this in some ways as I think about the early days of ART that many interested in HIV prevention may recall, where we first were able to offer a single imperfect but better than nothing drug, AZT, and then as AZT rolled out and provided extended quantity and quality of life for many individuals beyond the absence of options that they had before that, other drugs were also developed to make incremental improvements. And those drugs in and of themselves, and then, as we know now, in combination with each other, provided even a more powerful impact on the quality and quantity of life of people living with HIV. That is analogous to what we, kind of how we think of the broadly neutralizing antibody possibilities for HIV prevention, or for that matter, for treatment, and possibly even for cure, where we start with the best option that we have that still merits, evaluation in clinical trials, but we don't stop working on making improvements to that option. And that's what's happened in the broadly neutralizing antibody field. Even as the AMP study was designed to evaluate the idea, as I mentioned in one of my early slides, the idea of this new path forward, this really landmark opportunity to assess whether this idea of antibodies for HIV prevention really holds water or is in fact going to work. While we're looking at that question, we're also looking at, can we make this antibody better? Can we find other antibodies in other individuals who've been living with HIV, controlling their virus off of ART? And then can we take those antibodies and put them in combinations that might be even more powerful than one antibody alone? And can we take those antibodies and make them last longer so that people don't have to take them as frequently? Can we make them work even better with our immune system to kind of supercharge them in a way and their potential for prevention and treatment? So that is in fact what's going on. That is what you've heard a good deal about this week and will continue to hear about in upcoming weeks and months. Combinations of antibodies like BRCA1 that bind to the CD4 binding site of HIV, but also antibodies that bind to other parts of that protein on the virion. And can you put them together in, as you said, a cocktail to make them even more effective, to make them either more potent, meaning you don't need as much of the antibody to provide protection or treatment, or to make them more broad, meaning that they can provide protection against even more strains of HIV. Those are two of the primary intents of antibody development and combining them in these kinds of cocktails.
Great. No, thank you. And that's really helpful. And some of you may have seen that IAVI and the NIH announced their partnership to develop some of these new broadly neutralizing antibodies, just as you had mentioned, expanding both their breadth and their potency. So definitely a space to continue to watch. We did have a question come in about, is there participants who have reacted to the antibodies? And can the study team detect if a participant is using other prevention methods?
So great question. So in terms of whether participants have reacted to the antibodies, I think that probably speaks to the safety profile question. And that data will be coming out in September. But I can say that early data looks good from a safety perspective with this antibody. And we'll get into more detail about that when we actually are able to analyze the results. Right now, this study remains blinded, meaning that we don't know whether a given participant received lower dose, higher dose antibody or placebo. So we can't really compare that kind of safety data and know if they reacted to the antibody until we know if they got the antibody at all. So that's the kind of data that will come out and be available to all of us in September. But I will say early on, it looks like participants did well in this study from that perspective. Now, can the study team detect if a participant was using other prevention methods? Yes, we can, and we do. We ask participants if they're using other prevention methods. When they come into the clinic, we provide HIV risk reduction counseling, including explicitly encouraging them to use all prevention tools at their disposal. This doesn't hurt our study by any means. We can still detect whether the antibody itself added to any prevention or protection that may also have been helped along by other prevention methods. And then for some prevention methods, in addition to asking participants, are you using them, we can actually test participants for them. And of course, we only do this with their consent. This is part of the informed consent in the study. But we can look for example, whether they're taking pre-exposure prophylaxis or oral PrEP in the study and detect whether or to what extent taking oral PrEP may also have contributed to any prevention of HIV in this study.
Great, and I think that really speaks to this idea that we do need a toolbox, and that perhaps broadly neutralizing antibodies are one tool in that toolbox, but could be used with other things. Now, somebody asked, is there an opportunity for the body to produce these antibodies, without having a passive infusion?
Yeah, that's a great question. Some people do develop on their own naturally antibodies similar to this that are broadly neutralizing. Unfortunately, the number of people who develop these kinds of broadly neutralizing antibodies on their own is relatively small. And typically, the body develops these antibodies kind of too little, too late, meaning HIV comes in, the person acquires HIV, and then maybe 3 months later, 12 months later, 3 years later, they develop these broadly neutralizing antibodies, but HIV has already established a foothold by that time, and even has evolved to evade the antibodies that a person might naturally make. So it's almost as if our natural immune systems are in a losing race with HIV, which is, the benefit of being able to give somebody this antibody right off the bat is that they don't get behind HIV. They can actually stay, ideally, a step ahead of HIV in terms of prevention, and then in terms of treatment as well. So that's the idea behind these antibodies, and that's one of the ideas that the AMP study will kind of illuminate. Is that idea operant here? Is that working out?
So that's really, really helpful about this idea of to produce antibodies. And that's something that you said before, is that the antibodies this is sort of a natural, you could think about it as a natural prevention strategy. We all have antibodies. And on that line of questioning, is there antibodies to the antibodies? So is there a potential for, and maybe this is something that the data will obviously show us in September, but for BRCA1 for the body to produce antibodies against BRCA1?
Yeah, that's a great question. You're asking about a concept that we call anti-drug antibodies, or ADA. And I can tell you that we will be looking for that in this study. So I don't think that those results will be available in September, but down the line, they may be available in September. But we have looked for that in the earlier phase studies that I referred to with this exact same antibody, with BRCA1. And it was not, those anti-drug antibodies were not observed against BRCA1 in those earlier phase studies. Obviously, we can always learn more from more participants, and so we will be looking at that as well in the AMP studies.
Great. I know we're getting up to the top of the hour, but I do want to open it up to the line to just see if anybody has any questions that they would like to ask directly. Again, for those streaming on YouTube, you should feel free to write into the comment section and we can go ahead and pass those questions on now. Great. Well, given the timing, I'll ask sort of a final question that I have, which is, I know that we're highly, these results are really being highly anticipated by advocates, given that it's sort of a new, a non-drug form, like you said, it's sort of this idea of antibodies, a new prevention strategy. What should advocates be looking for? What should advocates be preparing themselves for when thinking about hearing these results?
Yeah, that's a good question. I think actually my sense in talking with you all over the course of, well, really the last several years, but even in particular in the last few months as we look forward to these results, is that you are exactly asking the kinds of questions for which you should find those answers in September, and you should listen for them. I think that you've got your eye on the ball, honestly, that you are thoughtfully asking questions about safety. Is this really a tool that we can have confidence in? Yes, we know that our bodies make these types of antibodies naturally, but it's crucial to look at how we respond to any drug of any kind that we'll give to other participants. And then I think that it will be useful to look at if this does in fact work, really how much is required. That is a crucial question. How much is required to provide prevention? And is that amount an amount that we can really realistically manufacture and launch into the public sphere? And then last but not least, I would invite people to look at these two cohorts. We know there are so many differences, both regionally and across genders and across ages, across all demographic measures. And I think that looking at how these results pan out across the two studies, as well as when we combine the data from the two studies, will be really very interesting to see. We need tools for everyone, not just tools for one demographic or another. And so that's one of the questions that I'm really most interested in looking at. And I think that the study design, the two-study design, the kind of sister-brother study design, or what have you, is a really nice, elegant way of looking at those questions and really getting at a truly global tool, as I hope this will be.
Great. Well, you've really left us with a lot of great questions and a wonderful charge as far as next steps and what to look out for in September, when we actually do get to dive into the data and hear more about the AMP trial and the results that come out. I just really want to thank you for your time today and your expertise and sharing that knowledge with us. We really do appreciate it.
It's my pleasure. I so appreciate y'all's interest, very much so.
Thank you so much, Shelley. Have a wonderful day. And to everybody on the line, thank you so much. Please, if you do have questions, please continue to email us and follow along. And we hope you've had a wonderful AIDS 2020. Have a great day, everyone.
Great, thanks. Bye.
Bye.